In this video, I'm going to give you a very cursory overview of viruses and bacteria. So a virus is a little infectious particle. It's little more than just genes in a box. It's a little bit of nucleic acid wrapped in a protein coat called a capsid. And in some cases, there's a lipid envelope around that. Now, viruses can use either DNA or RNA as their genetic material. They are acellular, so they carry an incomplete toolkit for life. These are far, far, far smaller than a cell. They're just like a tiny little particle, can't even be seen with a light microscope. They cannot replicate or metabolize on their own. They have to use a cell's equipment. So they are so tiny that they're not able to carry with them the tools for replicating, the tools for metabolizing, the tools for carrying out any of the 7 characteristics of life. So they have to hijack a cell in order to use the cell's equipment to do those things. So viruses are not considered alive, but they do have some lifelike qualities while they are infecting a cell. They're not alive, but they definitely occupy a gray area between the living and non-living worlds. So they replicate, but only when inside a cell and only if they can hijack the cell's equipment for replication. They can't harvest energy from the environment, but they can use the cell's energy that the cell has harvested from the environment, so on and so forth. They evolve, but only if they replicate inside of a cell. So they are definitely in this like in-between area between alive and not alive. I want to start by talking about bacteriophages. A phage is a virus, and a bacteriophage is a virus that infects only bacteria. So I know bacteria is in the word bacteriophage, but focus on the phage part. It's a phage or a virus that infects bacteria. So bacteria get sick too. And I'm going to focus on bacteriophages because their life cycle is simpler and more standardized. So here's an overview of the bacteriophage life cycle. There's 2 life cycles that they can choose from. So here's the bacteriophage. This is the little virus. It has kind of like a robot shape. It is kind of like an automated robot. So it has these automated features. It attaches to the surface of the bacterial cell and latches on. Once it latches on, there's this little like sharp unit that sticks into the bacterial cell's membrane. So it sticks through the cell's membrane and shoots its genetic material into it, shoots its DNA into it. So here you can see the bacterial chromosome. Bacteria have circular chromosomes. They're not linear like ours. So our chromosomes have DNA in a straight line, but all coiled up. Bacterial chromosomes loop back upon themselves, so there's no beginning and no end. So here's the phage DNA in light blue. Now, this virus is at a crossroads. It's left its protein capsid. This is its capsid that it leaves outside, by the way. Protein capsid left outside. Now, the phage is at a crossroads. It can either follow the lytic cycle or the lysogenic cycle. I'm going to follow the lysogenic cycle first. So in the lysogenic cycle, the phage inserts its DNA into its host's chromosome. It genetically modifies its host. Here we call it a prophage. You can see the virus is this segment here, but it's become one. Its DNA has become one with the bacterial DNA. And every time the bacteria copies its DNA and replicates, the prophage gets copied as well. There's that prophage in light blue right there and right there. So all the descendants of this parent bacteria will inherit the prophage. Now, sometimes that virus, that prophage, contains new genes that give the bacteria new qualities. Sometimes it creates a pathogenic bacteria when the original bacteria was not pathogenic. There are many diseases that are caused by bacteria that are not pathogenic on their own. Their own bacterial genome carries no pathogenic genes. The bacteria was infected by a prophage, and the prophage included genes to produce some toxin. So the prophage genetically modified their host and cause the host bacteria to create toxins that make us sick. An example of a disease that's caused by a bacteria that is harmless on its own and only pathogenic because of its prophage is enterohemorrhagic E. coli, also cholera. There are actually many of them. All right, so that's the lysogenic cycle. So now, this can go on for hundreds of thousands of years where this lineage is now permanently infected by this prophage. All of the descendants are now different because of the prophage. Or the virus can go to the lytic cycle. So once it infects, so we're going back to step 1, the phage injects its DNA into the bacteria, and now it's following the lytic cycle. What happens here is the viral DNA gets expressed by the bacterial host. What does it mean it expresses the viral DNA? So the viral DNA is, just like our DNA, code for protein. Proteins like the viral capsid and a bunch of viral attachment proteins. In the lytic cycle, the bacteria are going to read the viral DNA and produce everything that is instructed by that viral DNA. So basically now this bacteria is creating viral pieces. Bacteria reads viral DNA and creates viral parts. That means it expresses the viral genes. So now all these pieces of new viruses are being built inside the bacteria, and it's the bacteria that's building them. The virus has hijacked the bacterial DNA replication equipment and DNA expression equipment. So now these phages assemble, all the little pieces come together, and they burst out of the bacteria, and they kill the bacteria when they do this. So in the lysogenic cycle, the bacteria does not die. In the lytic cycle, the bacteria dies. Does not die. However, there is still some danger in the lysogenic cycle because at any point, you notice this arrow continues back to the start. At any point, this virus could change its mind. So it's been inserted as a prophage and it's been living as if it's part of the bacteria. But at any moment, it could revert back to the lytic cycle. It could pop out of the bacterial genome and move into the lytic cycle where it forces the bacteria to create new viruses and burst out of the bacteria. The kinds of things that will cause a prophage to pop out of its host's genome and enter the lytic cycle are going to be environmental stresses. If the virus gets the cue that the bacterial host is in danger, that the bacteria might die, it jumps ship. It says, this bacteria is not going to make baby bacteria anymore, so I can no longer be passed to a new bacterial generation. So instead, I'm going to pop out, make millions of virus babies, and destroy this bacteria before it dies with me inside of it. Okay, so it jumps ship and makes many viral babies as it does. So the lytic cycle is dangerous. Lysogenic cycle doesn't kill the bacteria directly, but there's always this bit of a threat that its prophage will revert to the more dangerous lytic cycle. All right, so bacteriophages are viruses that infect bacteria. In the lytic cycle, the virus destroys the bacterial genome, hijacks DNA replication and gene expression equipment to synthesize new viruses, which burst out of the bacterial host. The bacteria dies. In the lysogenic cycle, viral DNA is inserted into the bacterial chromosome where it lies latent. It divides only when the bacterial cell divides. It's inherited by all the bacteria's offspring, all of its descendants. It genetically modifies the bacteria, sometimes giving it new qualities. The bacteria lives until the prophage decides to switch to the lytic cycle. A lysogenic virus may live for many, many generations, thousands of years in a bacterial lineage. It can revert to the lytic cycle anytime it senses that the bacteria is stressed. The lytic cycle is the virus's way to make many offspring rapidly and then jump ship in case the bacteria dies. Jumping ship has risks, though, which is why many viruses decide to stay in the lysogenic cycle instead. During the lysogenic cycle, they make less offspring, but they're safer. Because Once the virus jumps ship, once it's outside the bacteria, what if it never finds another host? What if it floats around until it's degraded in the environment and it never finds another host to infect? I've linked to an animation so you can see the viral life cycle in action. Now, bacteria are very simple cells. They're basically just, a cell membrane with a single circular chromosome, which I'll draw in blue. They also have smaller circles of DNA called plasmids. So this is the chromosome, and these are plasmids. All the genes that the bacteria needs to survive are in the chromosome. So its chromosome has all the essential genes that it can't do without. The plasmids provide extra superpowers in a way. You can take them out and the bacteria will still survive, but the plasmids might provide little X-Men powers. So like resistance to certain antibiotics, or resistance to heavy metals, or tolerance to high heat. Things like that, little extra powers that the bacteria doesn't absolutely have to have, but which could be useful. The plasmids we call transposable elements. They're a little bit like viruses, and in fact, some of them came from viruses. So if you look back at the previous slide, the virus looks like a plasmid when it first arrives. Now, not all plasmids are viruses, but some of them are. We call them transposable elements because they are rogue bits of genetic information. Rogue bits of DNA. They are not actually under the control of the bacteria. So they can get moved from bacteria to bacteria. Now, this is a little bit of an overview that I have to give you in order to explain how bacteria trade genes with one another. Now, of course, bacteria replicate, so they can pass their genes to their descendants, just like you and I can. And that's called vertical gene transfer. But all of these are called horizontal gene transfer. So vertical gene transfer is when a parent passes its genes to its descendants. So parent, descendants, so daughters. This is vertical gene transfer. But bacteria can also move DNA around through horizontal gene transfer. And that's like, we can't do that. That's like if you liked some of my features and you wanted to have them for yourself, and I just popped out some of my DNA and gave it to you, and now you're more like me. Now, we can't do that, but bacteria can do that. So give genes to friends. Friend, bacterial friends. So these are all examples of horizontal gene transfer. So once new DNA gets into a bacterial cell by any mechanism, part of it can then integrate into the bacterial chromosome, okay? So it doesn't matter how the DNA is getting in, but if new DNA gets into the bacterial cell, the bacteria can integrate it into its own chromosome. So it can take DNA from outside of it and integrate it. So there's 3 ways that this can happen. The first is transduction. A viral capsid accidentally forms around bacterial DNA fragments and moves bacterial DNA from one bacteria to another. So if we think back to the previous slide, to the lytic cycle, you remember how the bacteria was forced by the virus to build pieces of the virus. So here's all these viral pieces, and then they came together inside the bacteria. Now, this is going to include viral capsid proteins, which I'm drawing here, but it's also going to include viral DNA. So we'll do viral DNA in burgundy. So the bacterial host that was infected by this virus made new capsid proteins for the virus and copied the viral DNA for the virus. Well, usually the viral capsid proteins come together around viral DNA. But sometimes, I've been doing the bacterial chromosome in blue, so sometimes they come together around pieces of the bacterial chromosome. Now, when these baby viruses burst out of the bacterial cell in the lytic cycle, killing it, most of the viruses are carrying viral DNA, and they go on to infect the next bacteria. But some of the viruses are carrying bacterial DNA. So they bring that bacterial DNA from one bacteria to another. So this viral capsid attaches to the next bacteria and injects it, and now these could be completely different species of bacteria. This is an accident, but the consequence is that genes get moved around from one bacterial species to another, not because they're hybridizing with each other, not because of sexual reproduction between species. No, because viruses are taking pieces of bacterial DNA from one species of bacteria to another. And one thing that can happen is that a species of bacteria that has evolved resistance to antibiotics, well, the resistance genes can be moved from that one species of bacteria to a completely different species that hasn't evolved antibiotic resistance yet. And so we did transduction. That's viruses moving the DNA about from one bacteria to another. The next one is transformation. In transformation, bacteria pick up naked DNA from the environment. Why is there naked DNA? This is just DNA that's floating around on surfaces, floating around in liquids. Why is there naked DNA floating around? Because things are decomposing all the time. So even in a bacterial colony, usually there's many bacteria living together in the environment, and they die sometimes, and then they decompose, and then their bits are just separated, floating about in the environment. So you can have some species of bacteria, I'm running out of space to draw here. Some species of bacteria, here I'll draw this bacterial chromosome in dark blue, and I'll draw the naked DNA in light blue. So there's little naked DNA fragments. Not all species of bacteria can do this, but some bacteria can absorb, bring in that DNA and add it to their chromosome. Again, this DNA that's been floating around doesn't even have to be from the same species. The species of bacteria that are able to perform transformation, they can pick up the DNA from any dead species of bacteria and then absorb their qualities and become more like them. All right, conjugation. 3. A plasmid, remember the plasmids up here? A plasmid codes for its own transfer from one bacteria to another. And this happens in a process that's a little bit like sex. It's not exactly sex, it's the closest bacteria have. So a bacteria with a plasmid can give a plasmid to a bacteria that doesn't have it. So let's use this drawing up here since I'm running out of space. The plasmid will provide the instructions for its bacterial host to form an organ called a sex pili. It's like a little appendage. This is called sex pili. Now remember, this isn't really sex. So this sex pili is basically just a narrow little tube that connects the donor bacteria to the recipient. And a plasmid moves through it. So the plasmid or a copy of the plasmid can move to the recipient. And now the recipient has that plasmid too. And again, this doesn't have to be performed between bacteria of the same species. So different species of bacteria can trade characteristics, trade genes with each other in this way. So these are the 3 types of horizontal gene transfer, and all of them allow bacteria to spread antibiotic resistance genes or really any other trait. And this increases diversity amongst bacteria. So bacteria reproduce clonally. And yes, they acquire some diversity through mutations, but they also acquire diversity through horizontal gene transfer, moving genes between different species of bacteria, trading genes between species of bacteria. And remember how important diversity is for survival. Remember, if everyone's exactly the same, then the environmental stress that kills one kills them all. And then you get extinction. But if everybody's a little bit different, if you've been trading genes with people with the bacteria from the outside, then everyone's a little bit different. Everyone has unique strengths, unique weaknesses. The environment changes. It's even more likely that certain individuals will have a trait that's useful in the new environment that allows them to survive. The next video will be on gene expression.